What is Cryo-E.T? No, it's not a frozen ET. Cryo-E.T stands for Cryo-Electron Tomography. It is a biophysical technique used to study very small objects and their surroundings, such as proteins and other subcellular structures. How small are these structures? Take this nuclear pore structure, for example. This is about 150 nanometers in diameter. In comparison, a strand of your hair is about 80,000 to 100,000 nanometers in width. The cryo part of the name indicates the use or application of very cold temperatures. Typically, this is done by plunging the sample into liquid ethane that was cooled by liquid nitrogen. This near instantaneous freezing of the sample allows for the formation of vitreous ice, or ice that doesn't form crystals. This is because ice crystals in the sample can interfere with what the scientists actually want to study. The electron tomography part of the name indicates the use of electron microscopes to form a tomogram. An electron microscope is a microscope that uses electrons to help us see these images. This is in contrast to a light microscope that uses light. Electron microscopes allow for a higher resolution than light microscopes because electrons have smaller wavelengths. This allows for a better resolution. Electron microscopes require putting the sample within a vacuum and passing the electron beam through it. Cryo-E.T generally uses transmission electron microscopy or scanning transmission electron microscopy to generate two-dimensional images. These two-dimensional images, or tilt images, can then be used to reconstruct three-dimensional models of the original sample, known as a tomogram. An important distinction between cryo-electron tomography and other cryo techniques like single particle cryo-EM is that cryo-ET is actually a sub-technique of cryo-EM, while cryo-EM typically refers to single particle analysis, or SPA. While SPA relies on taking 2D images of lots of particles cryo-fixed in different orientations and then pieced together computationally, ET focuses on taking lots of 2D images of a sample tilted at different angles, hence the name tilt series. These tilt series images are then reconstructed computationally into a 3D tomogram. This means that scientists need to be able to take, store, process, and analyze large amounts of these photographs. These leaps in storage and computing power have made it possible for scientists to take advantage of cryotechnology and electron microscopy to study important cellular and subcellular particles with ever-increasing resolution. Nowadays, single-digit angstrom resolution is possible with Cryo-E.T. However, look at all these other methodologies. What is the advantage of Cryo-E.T? Other methods, like single-particle cryo-EM or X-ray crystallography, can get similar or even better resolutions. However, Cryo-E.T allows for imaging of a more near-native state. For example, imaging of subcellular structures within the context of the cell can provide scientists with more information and a more nuanced understanding of cellular mechanisms. In contrast, X-ray crystallography requires samples to be grown in crystal form, and this can be really challenging since some particles will not readily form crystals. Single particle cryo-EM involves isolating and purifying the target protein or structure from the cell and other surroundings. Cryo-E.T allows scientists to study structures within a cell or tissue environment. So Cryo-E.T can be the best tool to study less static phenomena like lipid-lipid phase separation in cells. For example, in this paper by Huang et al., the scientists were studying membraneless organelles called GBPL, defense-activated condensates, or GDACs. To study these GDACs in cells, Cryo-E.T was probably the best choice. The scientists found that the GDACs had dense protein clusters from the reconstructed tomograms, which each required 51 tilt images. In another study, this one by Zimmerli et al., nuclear pore complexes that were previously thought to have much smaller pore diameters were demonstrated to have actually much larger pore diameters when they were in normal healthy cells. The previously observed nuclear pore complexes were in a more constricted state, which occurs in more energy-depleted cells. And the thinking is that the previously done images were taken after purification. And so the purification process might have caused the nuclear pore complexes to be more constricted when they were taken out of their native environment. For this study, the scientists made 178 tomograms. These two studies demonstrate the power of Cryo-E.T as well as the immense amount of effort Cryo-E.T requires. With so many moving parts, the sizable community of Cryo-E.T scientists have formed online and in various institutions to help troubleshoot issues and develop new workflows and analyses. More techniques are being developed which provide scientists with even more tools to study nature at angstrom level resolutions. For more information, please visit the following resources.